so far I recall, I think I completed the bacterial reproduction. Do you hear me?
Yes, sir.
At the last day, I think, last day I didn't take the class actually, due to some network problem. Now, up to what I completed actually, tell me, please.
Sir, you could only draw one small diagram of cyanobacteria and then the skin went black.
Okay. That means cyanobacteria, just to start from cyanobacteria, and then whether I discuss archaebacteria or not, I think.
No, sir. Only you draw a diagram of cyanobacteria.
Okay. Thank you very much. Now you see, what at your last day? Is a cyanobacteria. This is the cell membrane, and black one is the cell wall, and outer to cell wall.
ঠিক আছে
হ্যাঁ
মিউট
লিংক পাঠাইছি তো এসএমএস করে দিছি
আচ্ছা ঠিক আছে
সৌম্যজিৎ মিউট করো
হ্যালো
সৌম্যজিৎ মিউট করো
হ্যালো
Here we will get double-stranded circular DNA. The ribosome gas vacuole chromatophore. Ribosome is similar to bacteria, 70S type. I think I discussed the function of gas vacuole earlier. Chromatophore contains chlorophyll A, phycocyanin, phycoerythrin. So these are basic pigments. Phycocyanin is actually blue pigment. This is the red pigment and is the green pigment. In cyanobacterial photosynthesis, oxygen is evolved. Oxygen is evolved as byproduct. That means whenever cyanobacteria were evolved, from then onwards, oxygen started to add to the atmosphere. Because in bacterial photosynthesis, you know, oxygen is not at all evolved. So, this is the pigment, so photosynthesis, so that's why you know, as chlorophyll A, universal photosynthetic pigment, because this is the pigment found in both chlorophyll A, in both higher plants and even in the cyanobacteria. So they photosynthesize similar to higher plants. In their photosynthesis, glucose is synthesized along with the evolution of oxygen. Already I discussed about the gas vacuole provides a buoyancy to the organism. This is cyanobacteria. Ribosome is 70S type. Now, and these are cell walls. These are cell walls, mainly made of peptidoglycan. These are cell membrane made of protein, lipid protein. Now, I am coming directly to some filamentous cyanobacteria. Example, suppose nostoc. In filamentous cyanobacteria, you will get some specialized cell. So if it is the filament, then in this filament you will get so you see, these are the vegetative cells. They are thin wall and small, but this one is the thick wall and it is called heterocyst. And heterocyst is specialized for nitrogen fixation. You know, nitrogen fixation is even carried out in the rhizobium type of bacteria, but there are some differences between these two. You see, if you think about the rhizobium, it's a soil bacterium. From soil, so this is the leguminous plant. You will get some root nodules here. Root nodules. And in this root nodule, rhizobium enters. So that you will see, if you go to the cortex, you will get a number of cortical cells, and here you will get the rhizobium. It enters from the soil into the root nodule. The rhizobium possesses an enzyme nitrogenous enzyme. It has been nitrogen fixation, but for nitrogenous it requires an anaerobic environment. For its activity. That means in the root nodule, anaerobic environment is required to be developed. But there are errors. So how can it be developed? So to develop anaerobic environment, This is a leguminous plant. This plant synthesizes an enzyme, a chromoprotein called lag hemoglobin. So who is to synthesize the leguminous plant? Synthesizes a chromoprotein called lag hemoglobin, and lag hemoglobin acts as oxygen scavenger. Lag hemoglobin acts as oxygen scavenger. That is, it is to remove out oxygen from the nodule. As oxygen removed out from the nodule, so an anaerobic condition is developed there, so that nitrogenous can help in nitrogen fixation. This is what in the rhizobium actually is a bacterium. But the problem here is actually here nitrogen enzyme heterocyst it possesses nitrogenous enzyme. Okay. But lag hemoglobin is not found here. Is not found. If it is so, that in the heterocyst, as I said, an anaerobic condition is required to be developed. So how can it be developed? How can it be developed? So, for this, it has an inbuilt mechanism. The heterocyst has an inbuilt mechanism. What is this inbuilt mechanism? Mechanism is that the membrane of heterocyst is not permeable to oxygen but permeable to nitrogen. Look here, the membrane of heterocyst is not permeable to oxygen but permeable to nitrogen, and in this way inside the heterocyst an anaerobic condition can be developed. As a result of which is not required. So clear now how anaerobic condition is developed inside the heterocyst. So characteristics of heterocyst. What are the characteristics? First, heterocyst characteristics I am telling you. First, large, pale colored, thick walls, large, pale colored, thick walls, possessing nitrogenous and help in nitrogen fixation. So, possessing nitrogenous and help in what? Nitrogen fixation. So, this is what about the characteristics of heterocyst. Role already discussed. Now, we are proceeding towards the mycoplasma. So, I am to rub it out. double-stranded circular DNA. Ribosome 70S type. Cell membrane. Remember this thing, Mycoplasma lacks cell wall. It lacks cell wall. Cell wall is not present here. So this is what are the mycoplasma, and you need to remember one thing. Two terms are used. One is PPLO, that is Pleuropneumonia like organism. Now, causes pleuropneumonia in human being. Second term is pleomorphism. What is pleomorphism? Some microorganisms Some microorganisms can change their size and shape at different environmental condition and time. And this phenomenon is called pleomorphism. An organism exhibiting pleomorphism organisms exhibiting pleomorphism are called pleomorphic organisms. Example, mycoplasma. So this is what about the mycoplasma. Then we are proceed towards the Archaebacteria. So basic characteristics. Cell wall is made of non-cellulosic carbohydrates. In cell membrane, cell membrane. So they can survive in adverse conditions. Adverse conditions. So this is Archaebacteria in general. Then I have to classify Archaebacteria. Archaebacteria. Methanogens. Halophiles. Thermoacidophils. Methanogens, example, Methanobacterium. These methanogens, they are actually found in the rumen of cattle animals. Some cattle animals, some cattle animals, they have this methanol, they have actually they in their rumen. In cattle animal, the rumen is a chamber where fermentation of cellulose, actually in rumen, reticulum, omasa, that in the stomach, fermentation of cellulose takes place. And methanogens help in the fermentation of cellulose. Now, that's why after fermentation, carried to the intestine, and then actually through the intestine it is to be expelled or even from the body as fecal matter. That's why in the fecal matters also, we get the methanogens. There is cow dung in case of cow, which is that's why used in biogas production. biogas or gobar gas plants are now in different areas. In Tripura, it is very less. I think I don't know whether it is not. Actually, earlier there was 2-3 biogas plants in Tripura also. I don't know their existence now. But remember this thing. You see, in some Rajasthan and in those areas, you will get a lot of biogas plants or gobar gas plants where cow dung is used. And it is possible because in the cow dung methanogens are present, and methanogens can produce methane, and which is used as actually it's called gobar gas and may be used in cooking and lighting. So these are the methanogens. Halophils, high salinity. Normally, whenever any microorganism are placed in saline environment, then what happens? These organisms cannot survive there. This is because whenever any microorganism is exposed to saline environment, from their body cell, water comes out, there is plasmolysis occurs, and eventually the organism will die due to shrinkage. But that's why sometimes we add salt. And in case of halophils, they can tolerate high salinity. How? First of all, having there is high internal salt content. Internal salt content is very high. Next important point is devoid of sap vacuoles, and hence plasmolysis can be avoided. Third point, mucilage covering. It is to protect the internal environment. Fourth, presence of osmoprotectants. What are osmoprotectants? If it is a cyanobacterial cell, then suppose you will get in the wall some osmoprotectants. What these osmoprotectants are going to do? They are to allow some salts to enter inside to equilibrize or to maintain equilibrium between the halobacterial cell and the environment. That means whenever it is required to maintain equilibrium between the external environment and the halobacterial cell, osmoprotectants present over here they have been absorbing some salts from the environment to equilibrize them with the external environment. So this is what about the halophils. Example, Halobacterium. Then thermoacidophils. they can tolerate high temperature and high acidity. That's why they are called thermo for temperature, as they can tolerate acidity fields. That's why called acidophils. Now, how can they tolerate high temperature? They can tolerate high temperature due to the presence of branched chain lipid in their membrane. I am repeating it again. As I said earlier, due to the presence of branched chain lipid in the cell membrane, they can tolerate high temperature. Due to the presence of branched chain lipid in the cell membrane, they can tolerate high temperature. Is it clear to you? Secondly, they can tolerate high acidity because they have some specialized resistant enzymes who can act at strong acidic pH. So two things I said. Point one, in case of thermoacidophils, how can they tolerate high temperature? They can tolerate high temperature as they have branched chain lipid, in their cell membrane, which has them to tolerate this. Secondly, they can tolerate high acidity, because they have some special resistant enzymes who can act at strong acidic pH also. You know pH, strong, so one, they possess branched chain lipid in their cell membrane. They possess branched chain lipid in their cell membrane. Hence, they can tolerate high temperature. They possess special resistant enzyme that act at high or strong acidic pH. Now I am going to ask a question. Whom to ask? Parishra Chakraborty, do you hear me?
Yes, sir.
You tell me what is pH?
Sir, pH is the measurement to measure whether the solution is acidic or base.
Okay, so how to measure? How can you
So-
measure? Please tell me.
It can be measured through litmus paper also.
Litmus paper also. Okay. Then I am okay. Orko Prabha, you raised your hand. Tell me.
Sir, pH is the potence of hydrogen concentration in a solution.
Hydrogen concentration in a solution. আর একটু বলো, আর একটু বাকি আছে।
pH is the potence of
হ্যাঁ,
hydrogen concentration in a solution.
solution একটু ঠিকই আছে তোমার। Expressed in negative logarithm. Otonu Ray, you also raised your hand. Tell me. Otonu? Atanu De, Vinayata Datta, anyone?
Sir, the negative logarithm of gram molecular concentration
exactly
of
right. Actually,
gram.
concentration of hydrogen concentration in a solution expressed in negative logarithm. Hydrogen concentration of any solution expressed in negative logarithm is called what? pH. So I am going
pH.
to send you the ID and password again. Anytime it will be stopped, and then again you have to enter. Huh? So is clear now? So what is pH and strong? Suppose in case of a human being, pepsin can act as a strong acidic pH, almost at 1.2. When concentration of hydrogen ion increases, the pH value. So this is all about the bacteria. Now we have to start with virus, viroids, prions, lichen. Virus, viroids, prions and lichens. First of all, virus. What it appears like actually, an ideal diagram is I am going to show you. Protein coat called capsid. D- DNA virus. Another type you see again the protein coat. Capsid. This is RNA. So you can call it RNA virus, like polio virus. But in some RNA virus, so in case of influenza virus, this is the protein code. You will get 1, 2, 3, 4, 5, 6, 7, 8. So RNA. And this is called envelope. If it is an influenza virus, here you will get some structure. like this. So as capsid is there, around the capsid, not in all, around the capsid, in some virus, you will get an outer covering called envelope. And they are called enveloped virus, so it is influenza virus. So what are these structures called? You see, so this is one is hemagglutinin. This is called neuraminidase. So two subunit antigens are found, hemagglutinin and neuraminidase spikes. And in case of influenza virus, the hemagglutinin may be termed as H and M. They vary. Basically, in case of RNA viruses, basically enveloped RNA viruses like influenza, antigenic variation is found. What is found? Antigenic variation is found. It means that the virus is changing its structure frequently. And this is possible because actually it is easier for an RNA virus because you know single stranded RNA is less stable. Sometimes it is cut and remixed, and in this way mutation occurs. So that suppose influenza virus, the H, maybe sometimes it is H1N1 type, sometimes it is H2N1 type, sometimes it is H2N2 type. So everyone is different from the other. As it is highly mutating actually, that's why the vaccine against RNA virus like this, those where high antigenic variation is found, high antigenic variation is found, it's very difficult to develop a vaccine. Because suppose, what is actually a vaccine is going to, how is this, suppose polio vaccine. What happens to polio vaccine? That generally in case of polio vaccine, polio virus is taken. Every viral particle or every pathogen has two parts. One is called is antigenic part, other is called is disease causing part. Remember this thing, suppose pathogen you can write. Has two parts. Disease causing part antigenic part. By disease causing part, the pathogen multiplies inside the host cell. By disease causing part, the pathogen multiplies within the host cell, and by antigenic part, it is to trigger the immune system. Whenever any antigen enters the body, it has the ability to stimulate our immune system so that our immune system can produce an enzyme, produce weapon or antibody against them. So, remember this in case of viral infection, whatever the type of virus it is, No medicine is going to treat it. Sometimes some medicine given to check the viral growth, not to kill the viruses. Because viruses can only be killed by the antibodies produced in our body cell by immune system. Whenever we are suffering from flu, it may last for 7 days. Whenever we are suffering from measles, it may last for 14 to 21 days. If I am suffering from, suppose someone is suffering from mumps, again it takes almost 21 days. Why? All these are viral infections. All these are viral infections, and there is no medicine actually. You have to take rest only. What happens here? Depending on the complexity of the structure of the pathogen, our immune system takes some time for preparing the antibody, the weapon. If it is a simple type, it may be developed within 4 to 5 or 6, 7 days. And once antibodies are developed, they start destroying the pathogens and eventually we generally recover. In case of measles, it must require 14 to 21 days. That's why we have to suffer for these days. So in case of any viral infection, we have to depend on our immune system. If our immune system is strong, then it can fight against it, like it starts producing antibody quickly so that we can be recovered soon. In case of, so what happens here, suppose in case of polio, so whenever a vaccine is prepared, suppose polio vaccine is to be prepared, the polio virus itself is taken, it is treated in such a way that its disease-causing part gets destroyed. Once its disease-causing part is inactivated, then antigenic part remains same. So when it is taken through drops, our immune system gets triggered and the information regarding the structure can be stored in the memory cell. But as this pathogen has inactive disease-causing part, it cannot multiply inside our body, so no symptom is developed. This is what, and then in reality, when actually the poliovirus enters the body, and our immune system is already aware about it, as soon as the poliovirus enters the body, memory cells get triggered, activating B lymphocyte cell to start producing antibodies. So that without producing any symptom, the microorganisms eventually will die. This is what happens in case of vaccination. That's why we need to be vaccinated, to make our immune system aware about some deadly pathogens from earlier. Okay, but if our immune system is not aware of it, then what will happen? Then also antibodies will be produced, but it will take some more time. In the meantime, we have to suffer. And now suffering increases wherever their particles multiply very rapidly. If the particle multiplies very rapidly, then what will happen? Then these viral particles, then what will happen? I am telling you, if they multiply very rapidly, then what will happen? If it multiplies very rapidly, means it is going to attack the organ system. Once organ system is attacked, obviously eventually the person will die. This is what happens. So in case of influenza virus, what I am telling you, is they are changing their antigenic property frequently. So how can a vaccine can be developed? So vaccine is developed against H1N2. In the meantime, the next time H1N1 enters. So whatever the vaccine given to me is all useless. Again our body is to fight, start producing antibody against it. And till antibodies are not produced in the body, we have to suffer. This is what happened here. So RNA virus, you know, they have some of the, not all, in some RNA virus, they have the high degree of mutation. So they are changing their structure regularly. And hence it is very difficult to develop a vaccine against a particular, suppose it's the coronavirus, you know, it's a RNA virus. So it is changing its structure. So you know, in the meantime, almost, I think 13 strains are available. So against whom vaccine will be. So there are multiple copies to be developed. So this is the most important thing. Just a minute. হ্যালো, আমি ক্লাসে আছি একটু। হ্যাঁ বলেন।
আমি শুভাঙ্কিতার বাড়ি শুভাঙ্কিতার বাবা বলছি।
আচ্ছা আচ্ছা, আমি একটু ক্লাসে আছি, একটু ধরুন না 8 ঘন্টা পরে করলে ফোনটা।
আচ্ছা
হ্যাঁ হ্যাঁ ওকে ওকে ওকে। So engineering deviation is actually what is, so that's why in case of RNA virus, even though it's very difficult to treat actually. It is difficult to produce a vaccine against any RNA virus. This is because RNA is single stranded. It actually shows it can inhibit them, it can be bred because it is less stable and again remixes so that every time a new strain develops. And that's why it is very difficult. So this is not about the basic part of the viruses. Remember this thing, in virus, out of 2 genetic material, DNA, and only one is present at a time. That means either the virus contains RNA or it contains DNA. Normally, in all cellular organisms, from bacteria to human being, in everywhere, you will get everywhere you will get both DNA and RNA together. You will get both DNA and RNA together. But in case of virus, either you will get DNA or you will get RNA. One of them, not both. Secondly, whenever it enters the cell then actually it is to hire the cellular machinery. It doesn't have any cellular machinery. Only the nuclear material enters. It doesn't have the cellular machinery. So what will happen? So they have to hire the cellular machinery. How? Whenever they enter a cell, it is to inactivate the cell nucleus. So the cellular machinery is under the control of this viral particle. And it starts producing its own components. And in this way, from one cell, it is released out, enter a new cell, again released out, and enter again a new cell. And this process is going on. This is what about the viral particle. Is it clear to you? Or you have any question? Do you have any question, please?
Sir, what is H1N1 and H2N1? These things, sir.
Oh, okay. One question coming from Vineta. Yes, smallpox virus, chickenpox virus, all are actually viral particles. Vineta, your question, smallpox, chicken, all of the viral particles. Now your question. Who asked me? Please.
Sir, what is the H1N1 and H2N1?
Okay. Look one thing, entity. What I say, in the surface antigens, hemagglutinin. Other is neuraminidase. Hemagglutinin is expressed by H. Now, H is of variety types. Sometimes the H may be, suppose, H1, sometimes it is H2, it may be H3. Similarly, it is also different types. N1, N2, N3. So, due to the change in the mutation in the genetic material, in some virus, influenza virus, it is either suppose nuclear. So it is E. coli cell. E. coli means Escherichia coli, a bacteria. Now, it is attacked by bacteriophage. Bacteriophage first sits on the wall with the help of its spikes and tail fiber. You know, the structure of bacteriophage, they have the spikes and tail fibers like these. They are the spikes and they are the tail fibers. So spike it actually sits on it and tail fiber is wrapping around the bacteria. Now what happens? So we will get the DNA here. Then what happens next? From these parts, lysozyme is secreted. And so that a hole is created by the activity of lysozyme and phage genome enters the bacterial cell. Phage genome. The capsid part remain attached to the bacterial wall and it is of no existence. After entry, it enters either, I am showing you, lytic cycle or lysogenic cycle. So what happens in the lytic cycle? You see, swapping it out. This is the phage genome. It is to inactivate the whole genome. This is the whole genome, you can write. Nuclear and genome, same thing. So this step is the inactivation of host genome. After that, what will happen? Then cellular machinery got inactivated. Whole genome. Now the cellular machinery is under the control of phage genome. Cellular machinery is now under the phage genome. And phage genome. What happens next? Then what will happen? It starts producing its own components. Means it starts producing its own components. That means Head is formed. Neck is formed. Middle piece formed. Spikes are formed. Tail fibers are formed. Even first DNAs are formed. So, in this stage what happens? Synthesis of first component. Here you see they all are synthesized like non-living thing, like a computer, like a laptop, like the mobile machine. Different parts are produced. No living thing can reproduce like this. No living thing can reproduce like this. Then what happens? Here assembly of first component. Next is the assembly of first component. And here what happens? Files are formed to the nuclear material inside. Then lysis.
Sir, what you have written, cellular machinery is now under the
Control of first genome. Control of first genome.
Sir, I cannot see this word.
Cellular machinery is now under the under the control, okay, of
Yes, sir.
Is it
Yes,
clear
sir.
now?
Yes, sir.
Now assembly of first component and formation of daughter phases. Next, lysis of host cell and release of daughter phases. So what happens here? Daughter phases are released out. But in case of lysogenic cycle, what happens? The first genome gets attached to host genome. First genome gets attached to host genome. At this point, whenever first genome gets attached to host genome, it is called, this first genome is then called prophase. Whenever first genome is found to be present in prophase stage, it is not going to do any harm to the host cell. Then it starts dividing along with the host cell. So host cell divides to form two bacterial cells are formed. In every bacterial cell, you will get the genome. It further divides, suppose, forming 4 bacterial cells, and every cell you will get the first genome attached to it. Then, so in this way, a huge bacteria colony may be formed, but the bacterial cell is not at all affected. But if somehow the colony comes in contact with adverse conditions, somehow, maybe heat, maybe toxic chemicals, then what will happen? Half genome gets detached from the host genome. So what do you write? Half genome gets detached from host genome. You will make a snapshot also. That is easier for you. After that, what will happen? It enters the lytic cycle. It enters the lytic cycle. So, this part is lysogenic until first genome, and then you will get the lytic part. Is it clear now? In a lysogenic part, you will get up to this. Once it is exposed to some adverse condition, first genome gets detached from the host genome and enters the lytic cycle. So this part is lytic cycle. Here, take a photograph please. This part is lytic cycle and this part is lysogenic. Okay, one thing I am telling you now, I am not going to give you the exercise today. I will be meeting in the next month so far, and in the meantime you have to study. You can write me directly in my WhatsApp group, and in my personal WhatsApp, if you have any problem, I will be solving your problem. For this month, I have to stop this for unusual circumstances, and from next month, as usual, the class will be continuing. No problem. So, and I will take something like this so that there will be no problem in future. Okay. So, you have to follow the classes, you have to follow the class, take the books, and if you have any problem, please tell me. So then